I'm going to talk about a trial, a randomized control trial of a complex intervention, not a drug trial, called the SAFER-2 randomized control trial. And our objective was to evaluate new protocols for emergency ambulance paramedics treating older people following a fall. In the intervention group, the paramedics had protocols which allowed them to assess the person, leave them at home, rather than taking them to the emergency department, and referring them to a community-based falls service. So that was the intervention group, and the control group had care as usual, which was usually taking them to the emergency department, although we know that emergency paramedics do make these decisions and leave people at home without any ongoing care. Or patients sometimes refuse to travel to the emergency department. Our aims were to evaluate outcomes for patients at 1 and 6 months, and our primary outcome was a composite measure. It was a hierarchical outcome, which was made up of death or emergency admission to hospital or emergency department attendance or 999, those 911 calls in the States. So we analysed our primary outcome by a composite of those events. The setting was 3 ambulance services in the UK, 2 in England, 1 in Wales. This was a cluster randomized control trial. Patients were not individually randomized because we felt it was impossible for paramedics to switch on and off the learning of the new protocol. So we randomly allocated ambulance stations, so teams of paramedics, to either receive the training and the new protocols or continue with usual care, the control group. It was a large trial. There were 31 eligible stations across the 3 ambulance services. Sorry, this is not the best slide ever, but you can see at the bottom we ended up with 2,389 patients in the intervention group and 2,264 patients in the control group. So a very large trial. In this trial, we got the data for our outcomes from 2 routes. And some of the data we had in both of these routes, one being usual identifiable follow-up from hospitals, and the other was using routine anonymized linked data outcomes. So for emergency department attendances and hospital admissions, we had data through these 2 routes, which meant that we could actually look at the 2 routes together and check the quality of the data and look at the outcomes we got from each of these routes. So the route 1, following up identifiable data, is the traditional route that you would use in a randomized controlled trial. And the researchers visited the hospitals to access hospital notes and get these outcomes. This was about dates and current events of emergency department attendances and hospital admissions. And in route 2, we followed up using routine linked data that was non-identifiable from central sources. So you can imagine there were a lot of permissions that we needed to do to get to this point where we were able to do this. And the agreement with the ethics committee and the information governance committees was that if the patients didn't actively dissent, we could follow up anonymized linked data without consent. So our sample size then was, we aimed for 6,000 participants, 1,300 dissented from further follow-up. So we took those patients completely out of the trial. 4,700 patients consented to being followed up through identifiable data route, and 1,550 also consented to follow up via route 1. I'll explain a bit more. What we are interested in is the characteristics of the sample of the patients who identified to be followed up through 2 routes, because we could compare the quality of the data through those 2 routes on those patients. So data linkage in this sense is pretty new in the UK, and I think the UK is quite ahead of internationally. It's not an easy thing to achieve, so we're able to link data from different sources, data about deaths, data about emergency department attendances, and data about hospital admissions, without ever identifying the patients. So the clinical information is separated from the identifying information. Once these patients go into this route, you can never identify them again. We can never go back. We can never unpick it. In fact, in this trial, we were able to match 99% of our patients. So we had an extremely good linkage rate. So the patients that we had in both groups then were in England and Wales. Through the data are there, through the identifiable route, 1548 patients from the 3 sites, anonymized data only, 3,107, and you have the 3 sites there, and then overall the figures. And we looked at the characteristics of the patients at the call. We can straight away see that there are some differences in the patients that went through these 2 routes. So patients were slightly older that we looked up, that we were able to follow through the anonymized route. We had, the gender was pretty similar. The percentage that called out of hours in unsociable hours was higher in our anonymized route. And the distance from the ED was fairly similar, a little bit longer in the identifiable group. It's not the details I'm trying to get across here, it's more the fact that there were differences. The patients that we followed up in these 2 routes were different. So that in a traditional trial, what I'm trying to get across here is in a traditional trial where you follow up through identifiable data only, you may not get a complete picture of all of the trial participants. We wanted to look at, in terms of our outcomes, did we have, through these 2 routes, did we have the same dates of death, the same ED attendance dates, the same hospital admission dates, and the same hospital length of stay? So we've compared this data in our English sites. There were 1,118 patients in the 2 English sites, and you can see here that through the different routes, we had quite different results. So if we look here, through both routes, they agreed, whether it was the anonymized or the identifiable route, 622 patients had no further event. And 401, there was also agreement that there was a further event recorded. But in the anonymized route, we had 90 more events recorded than in the identifiable route. This is the same group of patients. They would imagine these data would match. When we look at hospital data on the same 1,118 patients, we have the same thing. We have more data. We are retrieving more data through our anonymized routine linked data than we are when we go to hospitals and retrieve that data manually. More events. For mortality, it's much more similar. So we didn't have such an issue with discrepancies between the mortality data through the 2 routes. When we look at our data components of the primary outcome, when we look at the proportion who had a further emergency department attendance, again, we find a similar picture. We find that we identified more emergency department attendances through the anonymized route than through the traditional identifiable route. So these, we don't know the reason for this. We can speculate, and we'll go on to that a little bit further. The same with the hospital attendants. Throughout these data, when we look at the anonymized route, and these are even for the same patients, and remember that we had way more patients in our anonymized route because lots and lots of people didn't consent to identifiable follow-up. So this is just about the people that we had in both routes. The data are not the same. And it's consistently more events in the anonymized data. So what we conclude from this is that subgroups of patients allowing identifiable data to be collected, the people that consented to have their data followed up, don't have the same characteristics as those people that didn't consent to that, but didn't dissent either, and we were able to do the anonymized follow-up. There was some agreement between our primary outcome through the 2 routes, but not perfect agreement. And our point here is that it's very difficult to do this at the moment, but in the future we think we'll be doing this more and more and more, and that you get both better coverage, better inclusion of patients through an anonymized data linkage route, and also more complete data than you do through the traditional route going to hospitals to manually seek out those records. It's far more labour intensive, and we're showing through this study that it doesn't identify as many events as the anonymised linked data route. Thank you. Did anybody want to ask any questions about that? Yes, Lee.
So, any speculation on why you get more from the, unidentifiable subjects?